Before moving on to the important points about amino acids, let us first understand what are amino acids. So in this video, we are going to have an overview of the structural features of amino acids, and also I will be providing a chart which mentions all the 20 amino acids which are commonly present in proteins along with their structures and the important points of each and every amino acid. Also, the link of that chart will be provided in the description box below. So what are amino acids? Amino acids are compounds where we have an alpha carbon, and to this alpha carbon there are connected 4 groups. Now these 4 groups are usually different in most cases, but in one single case the R group and the hydrogen group, they are same, which means that the R group is basically a hydrogen and nothing else, and that case is that of glycine. But apart from that, in most cases we have an R group, a hydrogen atom, a carboxyl group and an amino group attached to this alpha carbon. Now this R group can be as simple as that of hydrogen, which is the case in glycine, but it can be as complex as in the case of proline, where the R group is again bonded to the amino group of our alpha carbon. Now since all these 4 groups are different, so we can say that this alpha carbon is a chiral carbon. But what about glycine? No. In glycine, this R group is replaced by a hydrogen. That is why there are 2 hydrogens attached to the alpha carbon. So it will not be a chiral carbon. But in all other cases for amino acids, the alpha carbon is actually a chiral carbon. Now since that we have a chiral center with 4 different groups, except in the case of glycine, so we can say that most amino acids are optically active, which means that there will be 2 stereoisomeric forms, the D and the L form. Now let us take the simple example of alanine here. Here we have drawn the L form and the D form of alanine. If we look at them carefully, we will find that these 2 diagrams or these 2 figures are actually non-superimposable mirror images of each other. So therefore we call them enantiomers, the D and L enantiomers. The L-alanine is an enantiomer of D-alanine and vice versa. So that is all about the structure of amino acids. Now amino acids are basically zwitterionic structures because of the presence of separate negative and positively charged groups, and that is why they also act as weak acids as well as bases, and therefore they are known as ampholytes. So here we have a very interesting thing to note. Even the simplest of amino acids containing a single amino and a single carboxylic group, for example alanine, even alanine is diprotic, which means that each molecule of amino acid can donate 2 protons in aqueous solution. Now let us understand this a little more. So suppose here we have our amino acid in the fully protonated state and +1 charge is reflected on it. Why? Because hydrogen is neutral. Again, if we consider that this R group is neutral as well, then we can see in this fully protonated state the carboxyl group is neutral too. So the only charge that is available on this compound is this plus charge on the ammonia group or the amino group. So this plus charge is reflected here. Now in aqueous solution, upon increasing the pH, this hydrogen is released, which means that the carboxyl group leaves a proton. So this proton is given out. After this hydrogen leaves, the compound attains a 0 net charge. Why? because now this carboxyl group is not neutral. It has a negative charge, and this negative charge and this positive charge cancel each other, and we have a zero net charge on the compound. Now if we increase our pH further, then we will see that another hydrogen is released. Where from? Yes, from the amino group we have here. This hydrogen is released from the amino group upon increasing the pH furthermore. And finally we attain a negative charge, because now that this hydrogen is released, the amino group has acquired a neutral charge. So the only charge that is being reflected on the compound is that of this carboxyl group, and that is the negative charge. Now understanding this is very important, because this is mechanism occurring in titration of amino acids, which we will discuss in our next video. So here I have prepared a chart which mentions the important points along with the structure of the 20 amino acids which take part in translation. So you can pause the video and study the chart. Also, I will put the link to this chart in the description box below. With this, we come to our end. Now, let's do a quick revision. So, amino acids join with each other to form proteins. Therefore, they are the building blocks of proteins. Now, there are 20 amino acids which are commonly found in proteins. There are certain uncommon amino acids as well, but we are not going to discuss about that for now. Also, all these 20 amino acids are alpha amino acids, which means that the amino group and the carboxyl group is bonded to a single carbon, which is known as the alpha carbon. Again, in all the amino acids except glycine, the alpha carbon is bonded to 4 different groups. One carboxyl group, another is amino group, another one is our R group, and the other one is a hydrogen group. Therefore, we can say that amino acids have a chiral alpha carbon, excepting in glycine. Why? Because glycine is the simplest amino acid, and in glycine the R group is actually a hydrogen. Yes, it is nothing but a hydrogen. Therefore, we have 2 hydrogen in a single molecule attached to the alpha carbon. So we do not have a chiral center here, and that is why glycine is optically inactive. Now moving on to the next page. Because of the presence of chiral center, the amino acids are all optically active except glycine. Each of these optically active amino acids have 2 possible stereoisomers, which are non-superimposable mirror images of each other, and therefore they are known as enantiomers, the D, L and L enantiomers. Also, the amino acids have zwitterionic structure, which means that each amino acid has a separate positively charged and a separate negatively charged group. Next, amino acids act as weak acids as well as weak bases. Therefore, they are known as ampholytes. And the last point is very important because it explains titration of amino acids. This says that a simple monoamino and monocarboxylic alpha amino acid, which means that amino acid which contains only one amino group and one carboxyl group, and the R group of the amino acid does not contain any of the amino or carboxyl group. In that case, the amino acid will be diprotein, which means that it will be able to donate 2 protons per molecule to an aqueous solution when it is present in a fully protonated state. Now, the 2 proton donating groups in amino acid in a monoamino and monocarboxylic amino acid are the carboxyl group and the amino group, obviously. And that's all about the important points of amino acids. Now, there are some important properties of the various amino acids, and these are all taken from the chart that I have mentioned in the video. So I will just play the video. You can pause the video or take a screenshot whenever you wish to. All these points are mentioned in our chart as well. Now in these, the most important point here is that aromatic amino acids contribute to the spectroscopic property of proteins. We know that there are certain kinds of spectroscopy where we actually observe the absorbance of UV rays by the amino acids or by the protein. So that absorbance property comes from the presence of aromatic amino acids in the protein or the polypeptide chain. That's all. Thank you.